So that was heart rate, right? The influences of the autonomic nervous system, both sympathetic and parasympathetic, and a few hormones on our heart rate. Going back to our initial equation here, we also want to think about the other factor that influences cardiac output, which is stroke volume. So now we'll talk about stroke volume factors. So there are three main factors that can influence our stroke volume. One is the ventricular contractility, two is the end diastolic volume, and three is the afterload. So ventricular contractility is how hard those myocardial cells are working, how much force they're generating. That is going to influence how much blood is ejected. The second is how much blood has the ventricle filled, right? If we increase how much blood is filled into that ventricle, we're going to likewise increase how much blood can be ejected from that ventricle. So that will also influence our stroke volume. And then the third factor here is afterload. And afterload is this idea that we've got resistance in our distal vasculature. So if we've got a lot of resistance, if there's high pressures in our arteries, then it's going to be a harder, a higher force for the blood that's leaving the ventricle to overcome. And so if we have a high afterload, we're going to have a decreased stroke volume because the blood that's leaving the ventricle has to overcome the force or the pressure or the resistance that's in those distal arteries. Okay, so let's talk about these factors a little bit more in depth individually. So the influence of our ventricular contractility is how forceful we can expel blood from the ventricle. And that is also affected by the sympathetic nervous system. So the innervation of the sympathetic neurons to the myocardial contractile cells specifically is going to increase how hard these cells can work. And so we have the same chain of events initially. We have norepinephrine, either as a neurotransmitter or as a hormone, binding to our beta-1 adrenergic receptors. Cyclic AMP is, created as a second messenger. And then these four events are brought about, okay? We augment the opening of calcium channels. That means calcium channels are kept open for longer. We increase the calcium release from the sarcoplasmic reticulum, which means that more calcium can leave the SR and make its way out into the sarcoplasm. So remember, these are our two sources for calcium for contractile cell contraction, right? We've got calcium coming in from the extracellular fluid. So if we open up these channels for longer, more calcium can come in. We've got calcium coming from the SR. Again, if we keep those calcium pumps going on the SR, again, more calcium can make its way into the sarcoplasm. The third thing that we do is we increase the myosin ATPase rate. And that's how fast those myosin heads are making cross-bridge cycles. And if we increase how fast that can happen, we're also going to increase the force of contraction that those cells can generate. And then the fourth factor here, something we already talked about, if we can clear the cell faster, so if we increase the calcium pump on the SR, which can pump the calcium back into the SR more quickly, more rapidly, we can then create another sequence of events. So if we can relax the muscle faster, we can then contract the muscle faster. All right, and we're going to illustrate those a bit here. So if we've got, remember, the sympathetic nervous system is the only influence on our ventricular contractile cells, right? There's no parasympathetic influence. So we only have the influence of the sympathetic system on the force of contractility. So we've got norepinephrine or epinephrine, either as a hormone or neurotransmitter, binding to this beta-1 receptor, stimulating this G protein, turning on this adenylate cyclase, making the second messenger. And then what the second messenger does is 4 effects. All of these effects are in an attempt to increase how hard these cells are working. The first is it's going to keep these calcium channels open for longer. That's going to allow more calcium to come into the cell. The second is it's going to increase the rate of pumping the ATPase rate of these calcium pumps on the SR. This is also going to allow more calcium to move from the SR where it's being stored into the sarcoplasm as well. The third is it's going to increase the myosin ATPase rate of these myosin heads. That's going to allow them to make these cross-bridge cycles faster. The fourth is it's going to increase the calcium ATPase on the SR as well, but this is the active calcium pump that's pumping calcium back into the SR. And we talked about this idea, if we can allow the muscle to relax faster, we can then allow it to contract faster as well, okay? And so what we can see here is that if we look at the amount of force that a myocardial cell can produce at rest in a given amount of time, and we compare that to this myocardial cell under sympathetic influence, we see that the amount of tension produced is much more, and the amount of time that it can produce that tension is shorter. So a myocardial cell can produce more force in a shorter amount of time when it has that sympathetic influence, when compared to rest, that is, okay? And then again, I want to point out, very, very important here, there is no parasympathetic influence to the contractile cells. It's not significant. In fact, we completely ignore it. There's no significant parasympathetic influence to how hard those contractile cells work. And then we've got a few hormones here as well. So epinephrine, very same idea here. Epinephrine can be either a neurotransmitter coming from a neuron or a hormone released in the blood, and it's going to have the same 4 effects on those contractile cells. And then thyroid hormone, insulin, and glucagon are also other peripheral hormones that can have an increase in the force of contractility on our myocardial cells as well. And the mechanism here is not as important. We'll talk about glucagon a little bit in the GI system. Let's talk about the second factor that influences stroke volume. That's our end diastolic volume. That's how much the heart fills at the end of diastole. And so this brings into our interest the Starling's law. So the Starling's law says that if the heart stretches more and fills more volume, and those fibers stretch out as that chamber fills more blood, the fibers here get closer to their optimal length, and as they get closer to their optimal length, they generate more force, and as they generate more force, they can contract with a stronger force of contractility, and they can increase how much blood is actually ejected. So essentially, if I can fill more blood, I can eject more blood. I can increase my force of contractility and likewise eject more blood. And so what I want to point out here is a very important difference between the optimal length in skeletal muscles versus the optimal length here in cardiac muscles. So we talked about skeletal muscles usually being suspended in the body. So they're usually always at their optimal length at rest. And so we talked about the idea that if we increased or stretched the sarcomeres away from their optimal length, we would decrease the amount of force generated. If we contracted or squished the sarcomere together too tightly, we would also move the sarcomere away from its optimal length, and we would likewise decrease the amount of force that's generated. That was for skeletal muscle. Here in cardiac muscle, it's a little bit different. The myocardial cells, the sarcomeres, are not usually at their optimal length. The more that we stretch the sarcomeres, the more they approach their optimal length. And so it creates this law within the heart that if I can increase how far I stretch the sarcomere, how far I stretch these fibers, I can then let my fibers approach that optimal length, and I'm going to increase the amount of force that they can generate as they approach that, as they get closer to that optimal length. So the basic principle of the Starling's law is that if I increase venous return, and remember, venous return is how much blood comes back to the right side of the heart, I'm going to increase how much blood is filled into my ventricle, that end diastolic volume. And then I'm likewise going to stretch my fibers closer to their optimal length, increasing the amount of force that they can generate. And then I'm going to increase my contractility and increase how much blood is ejected as a result of that. All right, so here's an illustration of that. We've got our optimal length here. We've got the normal sort of resting state of our sarcomeres in cardiac muscle. And so as we see, when we increase the stretching of these fibers, and that stretching is being done because more blood is coming back into this ventricle. As I increase my end diastolic volume, I increase my stretching of those fibers, I increase the length of my sarcomeres. They're now approaching their optimal length. And so I'm going to increase how much force these cells can now generate, and I'm going to increase how much blood can be ejected from the heart, which is my stroke volume. Okay. And here's another illustration here. So for a given end diastolic volume, If we have an increased sympathetic activity, we're going to increase stroke volume. If we have a decreased sympathetic activity, we're going to decrease stroke volume. And so what this curve shows us, or what these three curves show us, is that even for a specific amount of filling volume, so end diastolic volume is one factor. We also want to think about the autonomic inputs here, so the sympathetic inputs specifically. For a specific or given end diastolic volume, if I've got a little bit of sympathetic input, my stroke volume will be low. If I've got a lot of sympathetic input, my stroke volume will be higher. And that's because I now have a harder, not harder, but a higher force of contractility. I'm going to squeeze more blood out of that chamber because those muscle cells are working harder. And so because of the sympathetic input, I'm going to increase my stroke volume, even though my end diastolic volume is remaining the same. Okay. Let's talk about the, so this is also so this is the final factor that affects stroke volume. So let's kind of recap here. We talked about the first factor, which is the end diastolic volume, and then we talked about the contractility of those myocardial cells. And then the third factor here is afterload. So we talk about afterload, we're going to kind of have to speak about preload first. So preload is the end diastolic pressure. It's determined by how much time the heart has to fill, how much pressure is in that atria, and then the central venous pressure. And central venous pressure is how much pressure is in the veins that are coming back to the right side of the heart. And so if we have a high amount of preload coming back into the heart, we're going to see a lot of blood coming back and filling into that chamber. The second thing we want to consider is afterload. Afterload is how much pressure is in the aorta or our distal arteries even during ejection. And we talked about this a little bit before. If we've got a lot of resistance in the aorta, if we've got a lot of resistance in our arterial circulation, it's going to be more of a force for the blood that's leaving that ventricle to try to overcome. So both preload, how much blood is coming back based upon the pressures in those vessels and the amount of time that the atria can spend filling, and afterload, how much resistance we've got in those peripheral vessels. Both of those factors will influence how much the heart can fill. Okay. All right, and then the final slide here is really a flow chart, which kind of connects everything together. The two factors that influence, so this is basically only stroke volume, I apologize. So this is only the factors that influence stroke volume and diastolic volume, how much the heart fills, the ventricle specifically, how much that fills, contractility, how hard those cells are working, and then the arterial pressure or the afterload, how much resistance is present in those vessels, in the aorta, and in those arterial arteries. And these are the three factors that influence our stroke volume. If we increase end diastolic volume, we increase our stroke volume. That is how much blood is filled. If we increase our contractility, how hard those cells are working, we will also increase stroke volume. If we decrease the resistance, right, the force that those vessels, that pressure has to overcome, we will then increase how much blood can leave the ventricle and move out into those arteries. Okay. So we don't have much time, unfortunately, to do any type of review, but I'm going to pause and see what specific questions you may have in the 3 or 4 minutes that we've got left. And if there are any burning specific questions on this topic, I can address those. Okay. All right, so I don't see any questions, but if you do have them, you can post them in the chat. In the meantime, let's look at this final poll question here. And let's look at some responses here. Oops, I apologize. Okay, let's go ahead and end this. So cardiac output decreases with decreased stroke volume and a heart rate. How do sympathetic and parasympathetic activity act? So a lot of responses for C, and C is decreased sympathetic activity decreases stroke volume, that's true, and heart rate, also true. And increased parasympathetic activity decreases heart rate only. Remember, increased parasympathetic activity does not have an influence on stroke volume. Okay, so the correct answer here would have been C. Let's kind of look at some of the others and explain why they're wrong. So A, decreased sympathetic and increased sympathetic activity, decreased stroke volume and heart rate. So this is not true, right? If we decrease our sympathetic activity and we increase our parasympathetic activity, we're going to, so decrease sympathetic is going to decrease the heart rate. So parasympathetic does not have an influence on stroke volume. So that's why this is wrong, I apologize. So decreased sympathetic and increased parasympathetic will decrease stroke volume, but increased parasympathetic has no influence on stroke volume. Okay, I hope that's clear. Option B, increased sympathetic activity and decreased parasympathetic activity decreases stroke volume and heart rate. Again, parasympathetic activity does not have an influence on stroke volume, okay? It does not influence the contractility of the cells, and so we can pretty much eliminate these two options. Option D says stroke volume and heart rate are unaffected by sympathetic and parasympathetic activity. We know that that's not true, okay?